Hello everyone, welcome to our study session 3. Today we're diving into the Tricarboxylic Acid Cycle. This is a fundamental part of cellular metabolism, and we'll explore how it functions and why it's so important. We'll cover the key steps, the molecules involved, and its role in energy production. I'm excited to guide you through this fascinating process. Let's get started. Welcome to this section of our presentation. We'll start with an overview of what you can expect to learn today. First, we'll introduce the main topics and set the stage for our discussion. Then, we'll define the key concepts of the TCA cycle, also known as the Krebs cycle, and explore its central role in energy metabolism. We'll look at the amphibolic nature of the cycle, meaning how it functions both in breaking down nutrients for energy and providing precursors for biosynthesis. Next, we'll discuss the anaplerotic reactions, which help replenish cycle intermediates, keeping the cycle running efficiently. We'll also examine the connection between the TCA cycle and diseases like beriberi, highlighting how deficiencies can impact metabolic pathways. Then, we'll give a brief summary of oxidative phosphorylation, the process that produces most of the cell's ATP. We'll review various inhibitors of the electron transport chain, which can disrupt energy production. For a more engaging learning experience, there will be tutor-marked assignments, either individually or in groups, to reinforce your understanding. We will conclude with a summary of the key points covered today and some self-assessment questions to test your knowledge, along with their answers. Finally, I'll recommend some references and further readings if you want to explore these topics more deeply. Throughout this presentation, I'll be connecting these concepts back to the overall theme of cellular energy metabolism and emphasizing their importance in health and disease. The citric acid cycle, also known as the Krebs cycle, was discovered by Hans Krebs in 1937. He was awarded the Nobel Prize for this discovery in 1953. Because of his work, the cycle was named after him as Krebs cycle, but it's also known as the Citric Acid Cycle or Tricarboxylic Acid Cycle. This cycle plays a crucial role as the final common pathway for breaking down fuel molecules, like proteins, fatty acids, and carbohydrates, into energy, carbon dioxide, and water. Without it, our bodies wouldn't be able to efficiently convert most of the food we eat into usable energy. Most of these fuel molecules are first metabolized into acetyl coenzyme A, or acetyl-CoA, or into intermediates of the cycle. The acetyl-CoA then enters the cycle and combines with oxaloacetate. During this process, 2 molecules of carbon dioxide are released, and the energy generated is stored in molecules called NADH and FADH₂. In the final stage, that stored energy is released and used to produce ATP, which is our main energy currency. So this cycle is fundamental because it links the food we consume to the energy our bodies need to function every day. After studying this session, I expect you to be able to describe the TCA cycle in detail, including its steps and significance. You'll also learn to explain the amphibolic nature of the cycle, which means it functions both in catabolism and anabolism. We'll cover the anaplerotic reactions that replenish cycle intermediates, ensuring it keeps running smoothly. Additionally, I'll connect this cycle to beriberi, a neurological disease caused by thiamine deficiency that affects the TCA cycle. We'll also give a summary of oxidative phosphorylation, emphasizing how it produces most of our cellular energy. Lastly, we'll look at examples of how the electron transport chain can be inhibited and what effects that has on energy production. Throughout, I'll highlight the key concepts, relate them to the bigger picture, and keep the discussion engaging and clear. In eukaryotes, the TCA cycle, also known as the Krebs cycle, takes place inside the mitochondria. That's because all the enzymes needed for this cycle are located within the mitochondrial matrix. This cycle is really important because it provides building blocks for synthesizing essential molecules like amino acids, purine bases, cholesterol, and porphyrins. Now, the cycle begins when a 4-carbon compound called oxaloacetate combines with a 2-carbon acetyl group from acetyl-CoA. This reaction produces a 6-carbon molecule known as citrate. From there, a series of reactions occur in a cyclic fashion. One key step involves converting citrate into its isomer, isocitrate, which then undergoes oxidative decarboxylation, meaning it loses a molecule of carbon dioxide. Next, the resulting 5-carbon compound, alpha-ketoglutarate, is also oxidatively decarboxylated, releasing another CO2 molecule and producing a 4-carbon compound called succinate. The cycle continues as oxaloacetate is regenerated through subsequent steps involving fumarate and malate. It's important to understand that oxaloacetate plays a catalytic role here. It helps initiate the cycle, participates in oxidation reactions, but is ultimately regenerated, ready to start the process again. And a major source of oxaloacetate is the carboxylation of pyruvate, which feeds into the cycle to keep it running. Overall, this cycle is essential not just for energy production, but also as a hub for creating key precursors for various biosynthetic pathways. Citrate is then converted to isocitrate through a 2-step hydration reaction catalyzed by aconidase. This process prepares the molecule for oxidation. Once we have isocitrate, it's oxidized by the enzyme isocitrate dehydrogenase, or IDH, to produce alpha-ketoglutarate. During this step, NADH is generated, and carbon dioxide is released. Next, alpha-ketoglutarate undergoes further oxidation by alpha-ketoglutarate dehydrogenase, or alpha-KGDH, forming succinyl-CoA. This reaction also produces NADH and releases another CO2 molecule. Then, succinyl-CoA is converted to succinate by succinyl-CoA synthetase, also known as succinate thiokinase. Interestingly, this step generates GTP, which can be used directly for energy. Following that, succinate is oxidized to fumarate by succinate dehydrogenase, and this reaction produces FADH₂. Moving on, fumarase adds water to fumarate, converting it into malate. Finally, malate is oxidized back to oxaloacetate by malate dehydrogenase, or MDH, producing another NADH molecule. This series of reactions completes the cycle, and as long as acetyl-CoA is available, the cycle can continue, fueling the cell's energy needs. Now, let's explore the dual nature of the citric acid cycle. When it functions as a catabolic pathway, its main role is to break down acetyl-CoA into 2 molecules of CO2, water, and energy. This process is especially important when the cell's energy levels are low, because in that state, the cell favors breaking down molecules to generate energy. The primary goal here is to oxidize acetate, capturing the energy released as reduced coenzymes, which are then eventually used to produce ATP. But the cycle doesn't only work this way. When the cell has enough energy stored, the same pathway shifts gears and operates as an anabolic pathway. In this mode, it provides essential building blocks for synthesizing various biological molecules needed for cell growth and maintenance. This flexibility is what makes the citric acid cycle so fascinating. It adapts based on the cell's energy needs. Because of this, we call it an amphibolic pathway, meaning it has both catabolic and anabolic functions depending on the circumstances. Understanding this dual role helps us see how interconnected and efficient cellular metabolism truly is. Basically, the TCA cycle has a single substrate, and that substrate is acetyl-CoA in most cells. However, there's a lot of withdrawal and addition of intermediates into and out of the cycle. This happens alongside the primary function of the cycle. These side reactions serve 2 main purposes. First, to provide building blocks for synthesizing compounds derived from various intermediates of the cycle, and second, to replenish the supply of intermediates as needed. This prevents the cycle from shutting down. For example, oxaloacetate and alpha-ketoglutarate are used in the synthesis of several amino acids. Citrate, on the other hand, is a key source of acetyl-CoA in the cytosol, which is then used for making fats, other lipids, and some amino acids. These processes represent some of the major ways the TCA cycle is drained for biosynthesis. This concept of adding back intermediates to keep the cycle running is what we call the anaplerotic nature of the TCA cycle. It highlights how interconnected and flexible this cycle is, not just for energy production, but also for biosynthesis and maintaining metabolic balance. Reactions that replenish the intermediates in the TCA cycle are called anaplerotic reactions. One key example of such a reaction is the conversion of pyruvate into oxaloacetate. It's important to understand that we don't always need to replenish the specific intermediate that's directly used in a biosynthetic pathway. Instead, replenishment can occur through a feeding-in process at any point in the cycle. For instance, when carbohydrates are being metabolized, the intermediates of the TCA cycle are replenished by the production of oxaloacetate from pyruvate. This highlights how flexible and interconnected the cycle is, allowing it to maintain its function even as intermediates are drawn off for other purposes. Understanding the anaplerotic nature of the TCA cycle helps us see how the cycle stays balanced and continues to supply energy and biosynthetic precursors efficiently. Beriberi, a neurological and cardiovascular disorder, is caused by a dietary deficiency of thiamine, also known as vitamin B1. This vitamin plays a crucial role in energy metabolism, particularly in the brain and heart. Sometimes, we see beriberi in alcoholics who are severely malnourished, which leads to a deficiency in thiamine. The disease presents with symptoms affecting the nervous system and the heart. Patients might experience pain in their limbs or abnormal sensations in their skin, which can be quite distressing. The heart itself can become enlarged, and if left untreated, the condition can progress to paralysis. Now, thinking about what's happening at the biochemical level, a deficiency in thiamine impacts several key processes, especially within the TCA cycle or the Krebs cycle. Since thiamine is a coenzyme for enzymes like pyruvate dehydrogenase and alpha-ketoglutarate dehydrogenase, its deficiency hampers the cycle's efficiency. This disruption affects how our cells produce energy, particularly in tissues with high energy demands like the nervous system and the heart. So, the connection between the TCA cycle and beriberi is fundamental. When the cycle slows down, energy production drops, leading to the neurological and cardiac symptoms we observe. Understanding this link helps us see how a simple vitamin deficiency can have profound effects on our overall health, reinforcing the importance of proper nutrition and metabolic function in preventing diseases like beriberi. Thiamine pyrophosphate is a crucial prosthetic group for two key enzymes in the TCA cycle, namely pyruvate dehydrogenase and alpha-ketoglutarate dehydrogenase. These enzymes are essential for energy production, as they help process nutrients into usable energy. In cases of beriberi, we see elevated levels of pyruvate and alpha-ketoglutarate in the blood, which indicates that these enzymes aren't functioning properly. This impairment is directly linked to vitamin B1 deficiency, which disrupts the normal functioning of these enzymes. Now, why does a deficiency in vitamin B1 lead to neurological issues? The answer lies in how the nervous system relies almost exclusively on glucose for its energy supply. Unlike other tissues that can switch to using fats through acetyl-CoA for energy, the nervous system depends heavily on glucose metabolism. This is where the pyruvate dehydrogenase complex becomes so important. It converts pyruvate, the end product of glycolysis, into acetyl-CoA, feeding into the TCA cycle for energy production. When thiamine deficiency inactivates this complex, the nervous system can't generate enough energy, which leads to the neurological symptoms we see in beriberi. So, in summary, the disruption of this pathway due to lack of thiamine hampers energy production in nerve cells, causing the clinical features of beriberi. This highlights the vital connection between proper enzyme function in the TCA cycle and overall neural health. Now, let's talk about the reduced coenzymes, NADH and FADH₂. NADH is actually the reduced form of nicotinamide adenine dinucleotide, and FADH₂ is the reduced form of flavin adenine dinucleotide. These coenzymes are generated during the TCA cycle, and their role doesn't end there. Once they carry electrons and protons, they themselves are oxidized, meaning they lose these electrons and protons. The electrons don't just disappear, they are transferred to oxygen, which acts as the final electron acceptor in this process. This transfer occurs through a series of molecules known as the electron transport chain, which is essentially a chain of electron-carrying proteins embedded in the inner mitochondrial membrane. As electrons move along this chain, a lot of energy is released. The key point here is that this energy is captured and used to produce ATP, the cell's main energy currency. This entire process of converting energy from electrons into ATP is called oxidative phosphorylation. So, in summary, the reduced coenzymes from the TCA cycle feed electrons into the electron transport chain, leading to a significant release of energy that is ultimately harnessed for ATP synthesis, powering many cellular functions. Now, let's talk about inhibitors of the electron transport chain and their practical applications. These inhibitors have been found to be quite useful as drugs and pesticides, especially when they are selective. For example, amytal is a barbiturate drug that can inhibit parts of the electron transport chain. Rotenone is a plant-derived compound commonly used as an insecticide, and it specifically targets the electron flow in insects. Additionally, there are antibiotics like pyricidin A and oligomycin, which block electron flow through the respiratory chain. By doing so, they effectively shut down energy production in their target organisms. This ability to interfere with electron transport has made these compounds valuable in medicine and pest control, highlighting the importance of understanding these inhibitors in the broader context of cellular energy regulation. All right, let's talk about anaplerotic reactions. These are really important because they help replenish the intermediates of the citric acid cycle that might be depleted during various metabolic processes. Without these reactions, the cycle could slow down or even stop, which would impact energy production. Some key examples include the conversion of pyruvate to oxaloacetate via the enzyme pyruvate carboxylase. This reaction is essential, especially when the cycle needs a boost or during times of increased demand. Another example is the transamination reactions that generate citrulline or other intermediates, helping to maintain the cycle's balance. Understanding these reactions is crucial because they link different metabolic pathways and ensure that the citric acid cycle can run efficiently, supporting overall cellular energy needs. Remember, these anaplerotic pathways are the cell's way of filling up the cycle's intermediates when they get used up or diverted for other purposes. Now, for your tutor-marked assignments, whether individual or group, I encourage you to explore these reactions further. Explain how they maintain metabolic balance, give specific examples like pyruvate carboxylase, and consider their significance in different physiological contexts, such as fasting or high-energy demand situations. This will deepen your understanding and help connect these concepts to the bigger picture of cellular metabolism. In this session, we have explored the Tricarboxylic Acid Cycle, also known as the TCA Cycle, and discussed its fundamental nature as a central metabolic pathway. We looked into how it connects with other metabolic processes and its role in energy production. We also examined its relationship with beriberi, which is a neurological disease caused by thiamine deficiency, a vitamin essential for proper TCA cycle function. Additionally, we covered the inhibitors of oxidative phosphorylation, which can disrupt ATP production and lead to various metabolic disorders. I am confident that you now understand the detailed description of the TCA cycle, including its amphibolic nature, meaning it is involved in both catabolic and anabolic processes, and its anaplerotic functions, which help replenish cycle intermediates when they are depleted. We also discussed how deficiencies, like in thiamine, impact the cycle and contribute to diseases like beriberi. Furthermore, we summarized oxidative phosphorylation, emphasizing its role in converting the energy stored in NADH and FADH₂ into ATP. We explored the different inhibitors of the electron transport chain, which can block this vital energy-generating process. Now, for your self-assessment, I encourage you to think about where the TCA cycle takes place, primarily in the mitochondria, and to describe how this cycle relates to beriberi, especially in terms of neurological effects caused by disrupted energy metabolism. Also, try to summarize the process of oxidative phosphorylation and explain how inhibitors interfere with the electron transport chain. Remember, understanding these key points helps connect the biochemical pathways to real-world health issues and overall cellular energy management. Hello everyone, and welcome to today's session. I'm excited to introduce our topic, Study Session 2, Generation of ATP in Glycolysis Pathway. Today, we'll explore how cells produce energy through glycolysis, focusing on the process of ATP generation. We'll go over the key steps involved and understand how this pathway fuels our cells. Let's get started. Welcome to our presentation. In this slide, we're outlining the structure of our discussion. First, we'll start with the introduction to set the stage for understanding glycolysis. Then, we'll look at the learning outcomes so you know what to expect and what you'll be able to do after this session. Moving on, we'll delve into the main content, which covers how ATP is consumed and generated during glycolysis, an essential energy pathway. We'll also examine how this process is regulated at different points, ensuring efficient energy production. Additionally, we'll explore some clinical conditions that arise when glycolysis is impaired, highlighting its importance in health and disease. After that, there will be tutor-marked assignments, either individually or in groups, to reinforce your understanding. We'll conclude with a summary of key points and then provide self-assessment questions and answers to test your knowledge. Finally, we'll share references and further readings for those interested in exploring the topic more deeply. This overview will help guide us through the detailed concepts ahead, connecting each part to the bigger picture of cellular metabolism. In this session, we continue our discussion of the glycolytic pathway. As you may recall, during this process, 2 ATP molecules are produced. However, it's important to note that even more ATP is generated when pyruvate is fully oxidized to carbon dioxide and water in the mitochondria. The regulation of glycolysis mainly involves 3 key enzymes that catalyze its irreversible reactions. Among these, the most critical control point is the enzyme phosphofructokinase, or PFK, because it catalyzes the first irreversible step that is unique to glycolysis. Now, once pyruvate is formed, it has 3 possible fates. It can be converted into acetyl-CoA, ethanol, or lactate, depending on the cellular conditions. Understanding these pathways is crucial because impaired glycolysis can lead to clinical conditions such as lactic acidosis or pyruvate kinase deficiency, which highlight how vital this process is for normal metabolism and health. After studying this study session, I expect you to be able to understand the gross and net ATP yield of glycolysis, which is fundamental for grasping how energy is produced in cells. We'll also go over the key glycolytic regulatory enzymes and their effectors, helping you see how this pathway is tightly controlled. Additionally, I want you to be able to explain the different fates of pyruvate, whether it enters the mitochondria for further oxidation or is converted into other compounds. We'll also cover some disorders of the glycolytic pathway, so you can appreciate how disruptions can lead to metabolic issues. Overall, these learning outcomes will give you a solid understanding of glycolysis, its regulation, and its clinical relevance. Let's take a closer look at how ATP is both consumed and produced during glycolysis. As you can see in this table, the process results in a net gain of 2 ATP molecules. It's important to understand that while some ATP is used up early on, the overall process generates more energy than it consumes. Most of the energy we extract from glycolysis isn't stored there. Instead, it's harvested later in the TCA cycle and the electron transport chain. These steps are where the real energy payoff happens, allowing our cells to produce the majority of ATP needed for various functions. So this table helps us see the balance, how initial investment in ATP sets the stage for a larger energy return in subsequent stages. It's a key part of understanding how our cells efficiently extract energy from glucose, fitting into the bigger picture of cellular respiration. The glycolytic pathway has a dual role in the cell. First, It degrades glucose to generate ATP, which is the energy currency the cell needs for various functions. Second, it provides building blocks for synthetic reactions, supporting the cell's growth and maintenance. The rate at which glucose is converted into pyruvate is carefully regulated to meet these two major cellular needs. If the cell requires more energy or building blocks, the pathway can be adjusted accordingly. Now, when we think about regulation, enzymes that catalyze essentially irreversible reactions are key control points. These reactions act as major gateways, so to speak, and their enzymes are prime targets for regulation. Specifically, in glycolysis, the reactions catalyzed by hexokinase, phosphofructokinase, and pyruvate kinase are virtually irreversible. Because of this, these enzymes don't just facilitate the reactions, they also serve important regulatory roles. Understanding regulation in glycolysis helps us see how the cell balances energy production with biosynthesis, ensuring everything runs smoothly according to its needs. Now let's talk about PFK, which stands out as the most important control point in glycolysis. This enzyme is tightly regulated because it essentially decides whether the pathway speeds up or slows down. Interestingly, ATP, our cell's main energy currency, actually inhibits PFK when its levels are high. It binds to a specific regulatory site, which is different from the active site where the reaction occurs. This means that when there's plenty of ATP, PFK activity decreases, slowing down glycolysis. However, this inhibition isn't permanent. When AMP levels rise, indicating lower energy availability, it reverses this inhibition and activates PFK, increasing the pathway's activity. So, the enzyme's activity is really sensitive to the ATP to AMP ratio. Lower ratios signal energy scarcity, prompting more glycolysis. Another key regulator is citrate. When citrate levels are high, it suggests that the cell has enough biosynthetic precursors. Citrate further inhibits PFK by amplifying the inhibitory effect of ATP, helping the cell avoid unnecessary energy production and precursor synthesis. It's also worth noting that enzymes like hexokinase and pyruvate kinase play roles in regulating glycolysis, especially at the committed step, the first irreversible reaction unique to this pathway. This step's regulation is crucial because it commits the substrate to glycolysis. Overall, PFK is most active when the cell needs both energy and building blocks. When energy or carbon skeletons are needed, the enzyme ramps up activity. But when both are abundant, PFK activity is almost shut down, helping the cell conserve resources. So, regulation of glycolysis, especially through PFK, ensures the cell responds appropriately to its metabolic demands. Now, let's take a closer look at how hexokinase and pyruvate kinase are involved in regulating the rate of glycolysis. Pyruvate kinase, specifically from muscle and liver, is allosterically inhibited by ATP. This means that when the cell has a high energy charge, or plenty of ATP, the enzyme's activity is reduced, effectively blocking the conversion of phosphoenolpyruvate, or PEP, to pyruvate. This feedback helps prevent unnecessary energy expenditure. Similarly, hexokinase is also subject to allosteric regulation, but in this case, it's inhibited by glucose 6-phosphate. When G6P levels rise, often due to a slowdown downstream, hexokinase activity decreases, preventing excess accumulation of G6P. Interestingly, when phosphofructokinase or PFK is inhibited, levels of fructose 6-phosphate, F6P, increase. Since G6P and F6P are in equilibrium, this leads to an increase in G6P as well. So, in essence, inhibition of PFK not only slows down glycolysis at that step, but also causes a rise in G6P, which then inhibits hexokinase. This interconnected regulation ensures a balanced control over glycolytic flux, preventing wasteful overproduction and conserving cellular resources. This coordination highlights how regulation at multiple points maintains overall metabolic homeostasis. The fate of pyruvate in generating metabolic energy actually varies quite a bit depending on the organism and the specific cell type. It's fascinating because pyruvate can be channeled into different pathways, leading to different end products. For example, in some microorganisms like yeast, pyruvate is converted into ethanol through a two-step process. This is a classic example of fermentation, especially under anaerobic conditions. In contrast, in muscle cells during intense exercise, pyruvate is often reduced to lactate when oxygen is scarce. And in most other cells, pyruvate is transformed into acetyl-CoA, which then enters the citric acid cycle to produce energy efficiently. So, understanding these different pathways helps us see how organisms adapt their metabolism to their environment and energy needs. A lot of energy in our bodies is produced aerobically, meaning in the presence of oxygen, primarily through the TCA cycle and the electron transport chain. Now, the starting point for this oxidative pathway is a molecule called acetyl coenzyme A, or acetyl-CoA for short. This molecule is crucial because it feeds into the cycle, enabling us to generate energy efficiently. Acetyl-CoA itself is formed inside the mitochondria, which are often called the powerhouses of the cell. The formation occurs through a process known as oxidative decarboxylation of pyruvate. So, pyruvate, which is a 3-carbon molecule, reacts with NAD+ and CoA to produce acetyl-CoA, along with carbon dioxide and NADH. This reaction is pretty important because it links glycolysis, where glucose is broken down into pyruvate, with the TCA cycle, allowing energy to flow smoothly through our metabolic pathways. The entire process is catalyzed by a complex enzyme system called the pyruvate dehydrogenase complex. Understanding this step is key because it highlights how our cells convert food into usable energy, especially in aerobic conditions. It connects the initial breakdown of glucose to the energy-producing steps that follow in the mitochondria, tying together the overall theme of cellular respiration and efficient energy production. This is the most common form of metabolic acidosis that we encounter. It typically happens when there's an overproduction of lactate, or sometimes when the body isn't using lactate efficiently. It can also occur if there's inhibition of pyruvate dehydrogenase, which is a key enzyme in energy metabolism. Additionally, this type of acidosis can be caused by rare congenital disorders where mitochondria fail to function properly, or in conditions like diabetic ketoacidosis. Liver or kidney disease can also contribute to this problem. Clinically, we see that lactate levels are greater than 5 millimoles per liter, and the serum pH drops below 7.35. Patients often present with symptoms such as nausea, vomiting, hyperventilation, which is the body's way of trying to compensate, and sometimes irregular heart rates. In essence, this form of lactic acidosis is closely linked to impaired glycolysis and energy metabolism, highlighting how disruptions in these pathways can lead to significant clinical consequences. Understanding these connections is crucial as we look at the broader picture of metabolic disturbances. So, here we're looking at a rare genetic defect in glycolysis that specifically causes hemolytic anemia. What happens in this case is that intermediates in the glycolytic pathway, especially those close to the pyruvate kinase step, tend to accumulate. At the same time, you'll see a decrease in pyruvate and lactate levels. This imbalance impacts the red blood cells' energy production, making them more prone to destruction. This increased destruction, or lysis, of red blood cells can lead to jaundice, which is due to the buildup of bilirubin from the breakdown of hemoglobin. The key point here is pyruvate kinase deficiency, which disrupts normal glycolysis in red blood cells, leading to their premature destruction and resulting in hemolytic anemia. This highlights how a single enzyme defect can have significant effects on cell survival and overall health. Let's wrap up our discussion on glycolysis by focusing on how ATP is generated during this process. Throughout this session, our main goal has been to deepen your understanding of the glycolytic pathway and its significance in cellular metabolism. Remember, glycolysis involves both the consumption and production of ATP. Initially, ATP is used to activate glucose, but as the pathway progresses, it produces ATP through substrate-level phosphorylation. This net gain of ATP is crucial because it provides immediate energy for the cell, especially in anaerobic conditions. We also explored how glycolysis is tightly regulated. Key enzymes like hexokinase, phosphofructokinase, and pyruvate kinase act as control points, ensuring the pathway responds efficiently to the cell's energy needs. This regulation is vital for maintaining metabolic balance and adapting to different physiological states. Furthermore, we discussed clinical conditions related to impaired glycolysis. For example, deficiencies in enzymes like pyruvate kinase can lead to hemolytic anemia, highlighting how crucial this pathway is for normal cellular function and overall health. To sum up, understanding the mechanisms of ATP generation, regulation, and associated disorders not only enhances your grasp of glycolysis, but also underscores its importance in health and disease. Keep these concepts in mind as they form the foundation for further studies in metabolism. Let's go over some key review questions. First, what is the gross and net ATP yield of glycolysis? The gross yield is 4 ATP molecules per glucose, but because 2 ATP are used up front in the process, the net gain is 2 ATP molecules. This makes glycolysis an efficient way for cells to quickly generate energy. Next, let's look at the glycolytic regulatory enzymes. These are crucial control points, including hexokinase or glucokinase, which catalyze the first step, phosphofructokinase 1, a major rate-limiting enzyme, and pyruvate kinase, which catalyzes the final step. These enzymes help regulate glycolysis based on the cell's energy needs. Moving on, what happens to pyruvate after glycolysis? Pyruvate can follow several paths. It can be converted into lactate under anaerobic conditions, which is important in muscles during intense activity. Alternatively, in the presence of oxygen, pyruvate enters the mitochondria and is transformed into acetyl-CoA to fuel the citric acid cycle, producing more energy. Finally, let's discuss some disorders of the glycolytic pathway. These can include enzyme deficiencies like pyruvate kinase deficiency, which leads to hemolytic anemia, or phosphofructokinase deficiency, causing exercise intolerance. Understanding these disorders highlights how vital proper glycolytic function is for overall health. So these questions help us connect the biochemical pathways to their physiological importance and clinical relevance, reinforcing how glycolysis fits into the bigger picture of cellular energy metabolism.